Hi, this little video is trying to explain meiosis. And in this example, we're going to use a cell that has 4 chromosomes or 2 pairs of homologous chromosomes. So here's our homologous pair here, and here's a homologous pair right here. And before meiosis can start, we need to have DNA replication, which happens before meiosis. So I'm going to make an exact copy of this DNA strand and an exact copy of this DNA strand, and similarly for this one, and similarly for this one. So now my DNA has replicated, but I still have 4 and 2 homologous pairs, and they tend to look like X's on a lot of the pictures when after DNA replication. And DNA replication happens right before cell division. So during prophase, and I remember this as PMAT, you have a nuclear, we're talking about a eukaryotic cell, and the DNA is inside the nucleus in a eukaryotic cell. And during prophase, the nuclear membrane disappears. So now you have the DNA, we're still inside the cell membrane. Metaphase 1 in meiosis, the homologous pairs pair up with each other. So I got this pair with the R's and S's next to each other, and this pair with the T's, A's, W's, and M's lined up with each other. And this is when crossing over can happen. So looking at these, so these are coding for the same types of traits. You might have crossing over where this little arm and this little arm are going to trade. They trade little arms. So now this one can go over there and this one can go over here. Now the advantage of that is that you have a new combination. So notice on this chromosome here, we now have a little t and a capital M, which was different than before. The little t was paired with the little m. So now we have a mix of all the traits that this organism's parents gave them. So these paired up. Now in anaphase, the homologous pairs leave. So now we're creating haploid cells at this point. And now in telophase 1, the nuclear membrane is reappearing. So now these become in one nuclear membrane, and so that gets the end of meiosis, where now of meiosis 1, and now we have 2 haploid cells. Now we move into meiosis 2, prophase 2, again the nuclear membranes disappear. Metaphase, they're lining up in the middle. Anaphase, these chromatids start separating. So like this when they were joined at the hip, it's a chromosome, and each one was a chromatid. Now the chromatids are separating during anaphase 2. And now in telophase, the nuclear membrane is reappearing. Now each of these, this is now creating 4, so each of these becomes its own nucleus. Now remember, meiosis is just looking at the nucleus. After this, now the whole cell has to divide. But now you got a nuclear membranes around each one of these. So now you have 4 at the end, and these are all haploid, because remember we started with a cell with 4, and now each one only has 2, and they're not paired. You notice the R and S looks different than the T, A, W, M. So these are not homologous pairs. It's haploid. It's only got 2 instead of 4. And now at the end of meiosis, we got 4 haploid cells. And I hope that helps with your understanding. Bye-bye.